Hello and welcome dear students. Today we are going to discuss an important topic of endocrinology, that is functions and mechanism of action of thyroid hormones. The main objectives of today's lecture are to get a brief idea about thyroid hormone action, to understand the mechanism of action of thyroid hormones, and then finally we will describe the various functions of thyroid hormones. To begin with, let us first discuss the mechanism of thyroid hormone action. Circulating thyroid hormones enter cells by passive diffusion via specific transporters such as the monocarboxylate 8 transporter, that is MC8, and monocarboxylate 10 transporter, that is MCT10, and an organic anion transporting polypeptide 1C1. After entering cells, thyroid hormones act primarily through nuclear receptors, although they also have non-genomic actions through stimulating mitochondrial enzymatic responses and may act directly on blood vessels and the heart through integrin receptors. Thyroid hormones bind with high affinity to nuclear thyroid hormone receptors alpha and beta. Both thyroid hormone receptor alpha and thyroid hormone receptor beta are expressed in most tissues, but their relative expression levels vary among organs. TR alpha is particularly abundant in brain, kidneys, gonads, muscle and heart, whereas TR beta expression is relatively high in the liver and pituitary, where it is associated with the negative feedback actions of the iodothyronines. Both receptors are variably spliced to form unique isoforms. The TR beta 2 isoform, which has a unique amino acid terminus, is selectively expressed in the hypothalamus and pituitary, where it plays a role in feedback control of the thyroid axis. The TR alpha 2 isoform contains a unique carboxy terminus that precludes thyroid hormone binding. It may function to block the action of other TR isoforms. The TRs contain a central DNA binding domain and a C-terminal ligand binding domain. They bind to specific DNA sequences termed thyroid response elements or TREs in the promoter region of the target genes as shown in the figure. The receptors bind as homodimers or more commonly as heterodimers with retinoic acid X receptors. The activated receptor can either stimulate gene transcription, for example myosin heavy chain alpha, or inhibit transcription, for example SH beta subunit gene, depending on the nature of the regulatory elements in the target gene. Thyroid hormones like T3 and T4 bind with similar affinities to TR alpha and TR beta. However, structural differences in the ligand binding domains provide the potential for developing receptor selective agonists or antagonists, and hence these are under investigation. T3 is bound with 10 to 15 times greater affinity than that of the T4 hormone. Although T4 is produced in excess of T3, receptors are occupied mainly by T3, reflecting T4 to T3 conversion by peripheral tissues. Greater T3 bioavailability in the plasma and the greater affinity of receptors for T3. After binding to the TRs, thyroid hormone induces conformational change in the receptors that modify its interactions with accessory transcription factors. Importantly, in the absence of thyroid hormone binding, the aporeceptor bind to co-receptor proteins that inhibit gene transcription. Hormone binding dissociates the co-repressors and allows the recruitment of co-activators that enhance transcription. The discovery of thyroid hormone interactions with co-repressors explains the act that thyroid hormone receptors silence gene expression in the absence of hormone binding. Consequently, hormone deficiency has a profound effect on gene expression because it causes gene repression as well as loss of hormone-induced stimulation. In addition to this generally accepted nuclear genomic mechanism of action, two other potential mechanisms could exist: a mitochondrial genomic action and a direct non-genomic mitochondrial action. The mitochondria are organelles within the cells which are associated with most of the energy provision of the cell in the form of ATP. They are also called as powerhouse of the cell, and they also have a small amount of DNA of purely maternal origin. There is evidence to support the likelihood that high affinity binding sites for the iodothioneins are present in mitochondrial membranes. And second, they influence mitochondrial metabolism directly by acting on mitochondrial DNA. In addition, there is evidence for a non-genomic action, and that the thyroid hormones can influence mitochondrial membrane transport processes directly once bound to the membrane located binding sites. One such suggested mitochondrial action has been the uncoupling of energy production from heat synthesis, and another concerns the exchange of intramitochondrial ATP for cytosolic DTP. Certainly, non-genomic mechanisms of action could account for those effects which appear to have a much shorter latency than those normally associated with T3 and T4. Furthermore, there may be plasma membrane TRs which mediate rapid, direct, non-genomic actions of thyroid hormones on ion channels and pumps. Dear students, now let us discuss in detail about the various functions of thyroid hormones. The principal actions of thyroid hormones, particularly T3, are metabolic in nature, but there are also important interactions with other hormones and regulatory systems. As discussed, most actions are genomic, but non-genomic effects are currently a source of much interest. Now, one among the functions of thyroid hormones is the basal metabolic rate, or BMR maintenance. One important function of T3 is the regulation of the basal metabolic rate in most tissues of the body, exceptions being principally the brain, spleen, and testes, as depicted in the figure. The basal metabolic rate is that rate of metabolism measured as oxygen consumption which exists when the fasted body is at complete rest in an environmentally neutral medium. Example, during sleep. T3 increases ATP turnover and utilization. A considerable portion of the basal metabolic rate is utilized simply for the maintenance of the cellular membrane pumps such as sodium potassium ATPase, which apart from anything else maintain cell volume. One major consequence of T3 stimulated basal metabolic rate is increased heat generation, that is thermogenesis. Thermogenesis not only is an obligatory component of energy expenditure, but also can be an adaptive mechanism against cold temperatures. A key tissue involved in this adaptive thermogenesis in babies is brown adipose tissue, that is BAT, and the mechanism involved in its regulation is the sympathetic nervous system in the presence of T3. At least part of the effect of T3 on BAT is due to an increased expression of mitochondrial uncoupling protein. Furthermore, T3 increases tissue responsiveness to catecholamines by increasing cyclic adenosine monophosphate generation, or CAMP generation, as well as interacting with CAMP dependent transcription factors to increase the expression of specific genes associated with metabolism. The clinical correlates are that hypothyroidism is associated with decreased heat production and the patient feels the cold, while the hyperthyroid patient generates a greater quantity of heat and becomes heat intolerant. Dear students, Let's now discuss another function of the thyroid hormone, that is protein metabolism. Many aspects of protein metabolism are influenced by thyroid hormones, and these relate to both anabolic and catabolic actions. These effects vary according to the tissue, the stage of development, and the level of the circulating hormones in the blood. Thus, during childhood, growth and development, the thyroid hormones increase the synthesis of proteins to a greater extent than the breakdown of protein. While in adulthood, this balance tends to be the reverse. Hypothyroidism is associated with decreased protein catabolism, while hyperthyroidism is characterized by protein loss, which contributes to the accompanying loss in body weight. The thyroid hormones influence protein metabolism mainly by acting via thyroid receptors as nuclear transcription factors, either stimulating or repressing gene transcription processes influencing protein synthesis. However, a non-genomic action on cell membranes associated with the stimulation of amino acid transport into the target cell has also been proposed. As an example of the temporal difference in T3 activity, hyperthyroidism in children is associated with enhanced linear growth, resulting in earlier fusion of the bone epiphysis, the end result being reduced height. At least part of this effect is due to the effect of T3 on specific TRs, mainly TR alpha 1 but also TR beta 1. In bone end plate chondrocytes, bone marrow stromal cells, and osteoblasts, resulting in overall stimulation of bone synthesis. For example, in bone matrix production. These receptors are also present in osteoclasts or bone eating cells, but the function of T3 in these cells is unclear. In contrast, however, hyperthyroidism in adults is associated with a bone resorption which is promoted to a greater extent than bone synthesis, so that there is an increased loss of bone matrix. This may be a reflection of greater effects of T3 on osteoclasts in adults. Dear students, now moving on to the carbohydrate metabolism. Dear students, as with protein metabolism, both anabolic and catabolic components of carbohydrate metabolism are influenced by the thyroid hormones T3 and T4. When normal circulating levels of thyroid hormones are present, the overall effect is glucose storage, that is glycogenesis, while higher, more pharmacological amounts such as in hyperthyroidism are associated overall with glycogenolysis, resulting in moderate hyperglycemia. For example, glycogen synthase activity is enhanced, increasing glycogen synthesis, while phosphorylase kinase, one enzyme which stimulates glycogenolysis, is also stimulated. The balance depends on the overall thyroid hormone levels in the blood circulation. Furthermore, in addition to direct effects on relevant enzymes, at least some of the T3 effects on carbohydrate metabolism are mediated by its interactions with other hormones. Thus, euthyroid subjects are sensitive to the effects of insulin indicated by the enhanced peripheral uptake of glucose into cells. Increased glycolysis and increased hepatic glycogen synthesis, while the catabolic effects of hormone such as the catecholamines, that is adrenaline and noradrenaline, are also enhanced when T3 levels are raised in the blood, resulting in increased glycogenolysis. The iodothyronines also promote gluconeogenesis indirectly by increasing the amounts of substrates such as glycerol and lactate. Dear students, let's now discuss about the role of thyroid hormones in lipid metabolism. The iodothyronines stimulate both anabolic and catabolic lipid metabolism, again depending on factors such as the stage of development and the levels of circulating hormones in the blood. Overall, the higher the levels of circulating thyroid hormones, the greater the breakdown of body fat, that is lipolysis, relative to fat synthesis, that is lipogenesis. Increased lipolysis is therefore associated with decreased levels of triglycerides and increased levels of fatty acids and glycerol. Cholesterol levels are also decreased with higher thyroid hormone levels in the circulation. Thus, in hypothyroidism, there is an increase in the fat storage, the body weight, while in hyperthyroidism, there is a decrease in body fat and hence body weight. Okay, dear students, let us now discuss the role of thyroid hormones in vitamin A synthesis. Because the thyroid hormones stimulate metabolic pathways, there is an increased demand for other molecules which play an essential role to the various metabolic processes such as vitamins acting as coenzymes. T3 is of particular importance for one such vitamin, that is vitamin A, because it stimulates its synthesis from precursor carotenes as well as its conversion to retinine. In hypothyroidism, the reduced synthesis of vitamin A can result in a build-up of carotenes in blood, which manifests itself as a yellowish coloration which can be observed in the skin. This is similar to the yellowish tinge commonly associated with the build-up of bilirubin in the blood, which is a sign of hepatitis. The two differ in that the sclera of the eye is spared from this coloration in hypothyroidism but not in hepatitis. Dear students, now we will discuss the function of thyroid hormone in brain energy balance regulation. Because of the metabolic effects associated with thyroid hormones, body weight is clearly influenced as seen when there is thyroid dysfunction. The increasingly common problem of obesity has focused attention on the possible additional roles that T3 may play in the brain regarding the regulation of energy balance. T4 and T3 can cross the blood-brain and blood cerebrospinal fluid barriers by means of two transporter proteins, that is OATP1C1 and MCT1, and hence reach specific target neurons within the neural tissues. Of particular interest is the widespread distribution of TRs within the hypothalamus, a region of the brain known to play a critical role with regard to the control of appetite and satiety. One crucial hypothalamic region involved in appetite regulation is the arcuate nucleus, and there are many thyroid hormone receptors here, implicating thyroid hormones in the overall control process. Dear students, now let us discuss the role of thyroid hormone in growth and development. It is clear that T3 and T4 play an important part in growth and development in utero as well as in the growing child and indeed the adult. The best way to appreciate the involvement of these thyroid hormones in these processes is to consider the clinical conditions associated with low and high circulating levels of these hormones in the blood. The most important growth phase occurs in utero and also in the growing child. Lack or absence of these thyroid hormones is manifested by abnormal, delayed growth and development seen following birth, a condition known as cretinism. The lack of growth and development, which is not only physical but also mental, becomes irreversible if treatment is not initiated as soon after birth as possible. Indeed, one of the first checks done on a newborn baby is the measurement of circulating T4 and TSH levels, because the condition is eminently treatable nowadays. In the adult, the lack or absence of T3 and T4 hormones is known as hyperthyroidism, and it too is characterized by an impaired metabolism and decreased mentation. That is a slowing down of thought processes which are characteristics of this disorder. The overactive thyroid, that is hyperthyroid state, is also associated in change in metabolism with aspects of increased metabolism becoming apparent, as well as change in cerebral processes with the patient becomes hyperactive and anxious. Okay, dear students, now we will discuss the interaction of thyroid hormones with the sympathetic nervous system. There are various interactions between the thyroid and its hormones and the sympathetic nervous system. For instance, increased sympathetic stimulation is associated with a decreased blood flow through the thyroid gland. This could influence follicular cell activity, for instance, by decreasing the endocrine regulatory signal provided by thyrotropin and the provisions of necessary metabolic products, for example, that of the iodide, for their normal functioning. T3 hormone acts as a transcription factor by stimulating the transcription of the beta-1 adrenergic receptor gene, thereby playing an important role in amplifying catechol-aminergic effects in specific tissues such as the heart, that is myocardial cells. Thus, in hyperthyroidism, the commonly observed tachycardia can be effectively controlled using a beta blocker. Dear students, now the effects of thyroid hormone on bone. Thyroid hormone transporters are present in chondrocytes, osteoblasts, and osteoclasts, as are thyroid hormone receptors, mainly the TR alpha 1 isoform. Hypothyroidism in children is associated with a delayed bone age and failure to grow, resulting in short stature if untreated. In contrast, hyperthyroid children have an advanced bone age, that is accelerated growth and premature fusion of the end plates of the long bones, also resulting ultimately in short stature if untreated. Thus, T4 and T3 are clearly involved in skeletal development. Similarly, in adults, hypothyroidism and hyperthyroidism are associated with decreased and increased bone turnover respectively. In hypothyroidism, both resorption by osteoclasts, that is bone reabsorbing cells, and remodeling by osteoblasts, that is bone forming cells, phases are lengthened, but the remodeling phase is affected more, so the condition is associated with an increased bone mass. In hyperthyroidism, both phases of bone resorption and remodeling are shortened since both processes are accelerated. Thus, in hyperthyroidism, there is a decreased bone mass and an increased risk of osteoporosis and consequently of bone fractures. Dear students, have you ever wondered that there is an influence of thyroid hormones on cardiovascular system as well? In addition to the indirect effects produced through interaction with the sympathetic nervous system, thyroid hormones directly influence several aspects of cardiovascular function. Both the contraction and relaxation phase of the cardiac cycle are accelerated in the hyperthyroid heart as a result of increased rates of release of calcium from the sarcoplasm reticulum and its reuptake. Opposite effects are seen in hypothyroidism patients. Thyroid hormones upregulate expression of the genes for the calcium channel, through which calcium is released during contraction, and the sarcoplasmic reticular calcium ATPase, by which it is retrieved during relaxation. In addition, T3 upregulates expression of the genes for the subunits of the sodium potassium ATPase and the potassium channel proteins that restore the membrane potential. Cardiac output is increased in hyperthyroidism and decreased in thyroid deficiency. So, hyperthyroidism lowers peripheral resistance by promoting relaxation of arteriolar smooth muscle and by increasing venous tone. As a result, venous return is increased, producing increased ventricular filling. The decrease in peripheral resistance also decreases resistance to ventricular emptying. These effects may be indirect, but when combined with the direct effects on cardiac myocytes and enhanced sympathetic actions, they have profound implications for cardiovascular function. Dear students, lastly we will discuss the function of thyroid hormones in nitrogen metabolism. Body proteins are constantly being degraded and resynthesized. Both synthesis and degradation of proteins are slowed in the absence of thyroid hormones, and conversely, both are accelerated by thyroid hormones. In the presence of excess T4 or T3 hormone, the effects of degradation predominate, and often there is severe catabolism of muscle. In hyperthyroid subjects, body protein mass decreases despite increased appetite and ingestion of dietary proteins. With thyroid deficiency, there is a characteristic accumulation of a mucus-like material consisting of protein complexed with hyaluronic acid and chondroitin sulfate in extracellular spaces, particularly in the skin. Because of its osmotic effect, this material causes water to accumulate in these spaces, giving rise to edema typically seen in hypothyroid individuals and to the name myxedema for hypothyroidism. Dear students, it was all about today's lecture. Hope you have understood it well. Goodbye. Thank you.